Ủa, Chiến là bạn khuyên Liêu này cũng nói tiếp luôn hả em? Đâu cái
And
sách.
dạ dạ
Could you
dạ
see my slides? Yes, yes, I can see the slide now. Can you make the full screen, please?
Okay. Okay, in the next part, I will have a brief introduction of our oncology solution. And first will be the guidelines for tumor NGAs and the solutions, and then the performance and some cases. And here is some guidelines and from the EMSO for the amplification for NGAs for oncology. And here is some clinic actionable for the non-small cell lung cancer for the EGFR or KRAS rate roles and for the MATE ERBB2. So, and for a novel target as NRG1. So these genes should be all covered by the panel. Um, and NCCN also recommended these genes should be covered in the non-small cell lung cancer. And for the advanced colorectal cancer, the KRAS, BRAF, MSI, KRAS, ARBB2, and the PLE should be covered. And the TMB, HER2 also recommended by the NCCN guidelines. And this also is the NCCN guidelines recommended KRAS, NRAS, BRAF, HER2, and NTRK and the rate. So these targets should be covered, even the small panel should be covered, all these targets. And for the ovarian cancer, the HRD is recommended as well as the fallopian tube cancer, the primary peritoneal cancer. This cancer HRD is recommended to be detected and to use for the PPARI treatment. And for some tumor agnostic genomic alterations, uh, a tumor agnostic means the pan cancer means all the solid tumor could be used, such as the NTRK, the MSI, the RATE, the BRAF, the FGFR123, and the TMB. But the TMB is not recommended measurement before we deciding to use the nivolumab plus the ipilimumab regimens. So if we do not decide to use this treatment, can be maybe not recommended. And here's the oncology solutions workflow I have showed before, both automatically and manually is supported. And here is our solid tumor panels for the 30 genes, 70 genes, 170 genes for all the solid panels, small panel to cover all the targets, therapy targets, and a large panel of 580 genes. All these panels could detect for SNV, indel CNV, and SV fusions, and MSI, and FFPE blood, and some other liquid could also be supported. And here is the data volume needed, and all could be automated. And we had added some special design for the rows fusion partners, such as CD74, SDC4, SLC 3482, and EZR regions. And the CMV, we could detect as low as 2.3 copies. And here we had contained the classic MSI set size recommended by the guidelines, and we had more than 100. Actually, we had 30 hundreds of the MSI sites for MSI detection, and we had SMP for the cross-contamination quality control. Here is the automation systems for the library building, the DNA extraction, and the quality control and the pooling. In this machine, it is one machine could do the extraction, library preparation, and the QC and the pooling. All this could be done in this machine. And all this machine could be easily connected with the LIMS. And he's compatible with a variant, a broad range of reagents. And here is our sequencer platforms fully by ourselves. And FastSeq 300 is very fast. It's very suitable for the short turnaround detection. And the GeneMind M, a little bit big throughput, and the SubSeq 5000 has very high output, and it's suitable for WES, WGS, and some big panel. And the SubSeq Q, we had very huge output, and it could be used for the very big cohort research and a very big amount of samples. So for SubSeq 5000 and for SubSeq Q, they had very high data quality as Q40. And for these two platforms, it has Q30 over 85%. Of these platforms are suitable for cancer mutation detection. And also we had AIs for fully automatically integrate reporting, and all the pipeline and the reporting could be done automatically, or also could be corrected manually. And here are some details for the panel. The first is a small panel for lung cancer and the colorectal cancer. And we see we had these three platforms, different numbers of samples. These samples, this number is for tissue or for white blood. If we want to pair and we need pair samples, the will be half of the numbers. And for this panel, we designed for lung cancer and colorectal cancers. We had 32 genes, and we had 12 fusion targets and more than 100 MSI sites. And we also include some chemotherapy targets of more than 50 genes comes from GKB database. And we had a detection of limit of 0.2% for ctDNA and 1% for tissue. Here the performance, the sequencing performance of the panel, we had a very high mapping ratio, nearly 99%, and we had very high on-target rate of near 80%, and the coverage uniformity is also very good. We had very high of 0.1 for mean coverage. Here is the detailed targeted list, and we covered the targeted list I mentioned in the guideline before. All these genes have been covered. Also, we had covered some other genes may have the potential for therapies. Here's the demo report. This report could be fully automatically output, and we had the clinical information, And we had the result summary. We had 5 somatic mutations. We had 1 copy number variation, and we had 1 gene fusion. Also, we had a being gene. I will introduce for this gene in the next slides. And the MSI status. And here the details of the variant, the therapies, and the potential therapies, and the class. So this would be the recommendation for the therapy, and the potential maybe if we lack of the therapies recommended here, we could take this into consideration. And this is the mutations description. So, and this is the copy number variation, this is fusion, and here's the MSI. Another is similar to the 30 gene panel, but is a little bit big, and over 70 genes is covered. It covered 76 genes with 15 genes fusion and MSI genes of more than 100, and also the chemotherapy targets. And the data and the sample could be supported by our different platforms. So it has a high sensitivity as then as the small panel. And we had also the MSI, and we could have some probes for the cross-contamination identification. And here's the on-target rate. We could see that it's also very high, and the coverage uniformity is also very high. Here's the detailed gene list of the lung and common cancer 70 gene panel, and this is the genes covered by all exon, and this chemotherapy targets, and some intra coverage. This intra coverage, as I mentioned before, is designed for the detection for the fusion, and the MSI sites recommended by the guideline and some other MSI guidelines. So the demo report is similar to I introduced before, so I just skip it. And we also had pan cancer 130 genes. This panel is designed for patients with solid tumor. All the targeted recommended by the guideline will be included. As I introduced in the background, the agnostic actionable targets all covered, except for the TMB and the HRD. And we have new 700 KB of the panel size, and we have different numbers of samples supported by different platforms. Also, these panels could be analysis locally, and the reports could be generated automatically. All the solid path cancer are supported by this panel. And here's the performance of the panel. We could see that the mapping rate on target rate and the coverage uniformity is very good. And here is the detailed gene list. We had additional coverage for other solid tumor and the intron coverage for the fusion detection. And the report is similar to the previous report. And we have a big panel of pan cancer. In this panel, we had over 580 genes. We covered TMB, MSI, and all the solid tumor is supported. And also the risk of mammalian cancer could be evaluated. So the FDA and NMPA approved all the drugs of solid tumor is supported, and the MSI, TMB, and MR, and PDM1 or some hyperproliferation genes are covered. For this panel, it's relatively big. It's more than 2 MB for TMB detection. And for the fast-seq, we only, for FCM, the array, we only support for 4 samples. And for subseq or GeneMind, more samples will be supported. So he covered a broad range of the variants, and the LOD is for tissue is also 1%, and the LOD for ctDNA is also 0.2%. The mapping rate and the coverage uniformity is very high. And here is the HRD panel. As I'm introduced before, the HRD, we need to detect for the LOH, TRA, and the LST. Also, we had added the solid tumor target for all the solid, such as the MSI and the fusion targets of all the solid tumors. So this panel will be some bigger than the other panel is over 4 MB, and tissue, especially paired tissue, is recommended. We need to be more than 8 gigabase for HRT detection. And here is the sample supported by different platforms. For large amount of sample, we recommended the subseq 5000 for the HRD detection. And here is the HRD report. Is similar to the previous report, but with some description of the HRD. And here the MRD detection, we use the WES or some customized WES for HRD. For whole exome sequencing, we should have more than 35 gigabase, and for the small or optimized WES, we need more than 15 gigabase for each sample, and for white cell, we need 3 gigabase and 7 gigabase. Also, we have covered all the SMV, Indel, MSI, and the fusion and chemotherapy targets. For this panel, fast-seq 300 should use the biggest array, and for the GenoLab N and the subseq 5000, more samples are supported. But for MRD, if we have big cohort of sample, the subseq 5000 is recommended. And here is the MRD detection workflow. We had the block draw before surgery, and the MRD for the blood pre-surgery, and we had tissue and the blood in. Then we do the WES, and we do the MRD for the blood post-surgery, and after treatment, we do the MRD surveillance, and we do the follow-up 3 to 6 months. And we need a 20 milliliter blood by cfDNA blood tube. This is a demo report, and we could see before. Here's the description of the patient, and here's the MRD summary. The baseline, the first time of MRD detection, the second time of MRD detection, and any other detection of MRD of the follow-up. And this picture of follow-up will be drawn. And also the clinical action the annotation of the variants will be outputted, and as the variant de details, the therapies recommended, and the class. So let's see some performance. The first, we use some reference materials for the performance verification, and we use the standard substance 1 and the standard substance 2 of these variants. These are some variants that often being detected by in the solid tumor, and we had a broad range of the frequency. For these detection, we have validated the detection on different platforms of GeneLab M, FastSeq 300 and SubSeq 5000. We could see that all these mutations could be detected. And we had validated our panel by some NMPA or Chinese FDA approved kit. As we could see, we are validated by the L858R and the 19-del, the ERBB2 insertion, and the ORC fusion, and the ERBB amplification. This was validated in the lung cancer, and this validated in the breast cancer. As we could see, our panel had a concordance rate of the 100% compared to the Chinese FDA approved kits. And the detection rate for lung cancer, we have over 5,000 samples. And for EGFR, we have 50%. And for the tissue, we had 53.8%. For the ctDNA, we had 41.3%. And from the expert consensus, we could see that the overall ratio of the EGFR mutation is around 50%, while the 19-del compromise around 45%, and the L858R compromise around 40%. And our detection rate is very similar to the detection rate mentioned in the expert consensus. And the detection rate for the colon cancer, we could see that the overall of KRAS mutation is around 40%, reported by some big cohort, and our detection rate is very similar to the big population data. And here's the TMB. The TMB, we need to construct some comparison between the WES and the panel. And we do some regression, and we could see that in the lung cancer and the colon cancer, the correlation between the panel and the exome has good correlation. This is a picture from FDA approved panel of PGDX helical tissue. And for HRD, we had validated for some samples from BRCA positive. And we could see that BRCA positive samples had all very high HRD scores or positive. Here is an example of the positive. And here is an example of the negative. So all the HRD positive caused by the BRCA could be properly detected. And for MRD, we had tests for some standard substance of low VIF of 0.02, and we could see that all these mutations could be detected by our MRD panel. Here are some clinical cases, and this is a diagram of the resistance of EGFR TKI. As I see the mechanisms of the second generation EGFR TKI would be the primary resistance and the acquired resistance. The primary resistance could be the EGFR exon 20 insertions and the being deletion polymorphism. And the acquired resistance could be some altered EGFR signaling pathway, such as the C797S mutation and some other mutations. So in this case, we use the liquid biopsy for the monitoring of some drug resistance. And in this patient, he received liquid biopsy after chemotherapy, and we could see the VIF of the EGF PL858R and the TP53. After chemotherapy, the cell-free DNA VIF is not very high, and after 4 months, he had progression, and we could see that the VIF of this variant increases. And then he received osimertinib, and it works. We could see the VIF of mutation decreases, and then we do the surveillance, but he got progression, as we could see that the VIF increases. And very importantly, he had been deletion, and he had RB and TB53 commutation. So, and based on the historical picture, he may have the small cancer cell transformation. And the beam depletion is associated with the primary resistance to the EGFR DKI. As we see from this picture, if the patient had BIN depletion, he had worse survival. And the EGFR RB1 and the TP53 mutations is associated with worse survival. As we could see in this picture, if the patient had these three mutants, they had the worst survival. And for this patient, finally, he had no no drug available. So finally, unfortunately, he got progression. And for case two, in this case, he had tissue to be sequenced by our lung and common panel, and he had a 19 Del. Then he received gefitinib, and he had resistance. Then he had ctDNA liquid biopsy. They found some other variants. Then he received osimertinib, and he had resistance. And ctDNA and found some heterogeneity. So let's see the detail. Firstly, he had 19 Del, and he could receive for the gefitinib. Then he got resistance, and we found he had amplification of EGFR, and he had very complex spectrum of this variant. And we could see this T790 and L792H, and these variants accumulated in this location. So based on these findings, we had these mutations are very frequently to be seen associated with the osimertinib drug resistance. And based on the variant we found in our results by liquid biopsy. We have drawn the heterogeneity or evolution tree by this. He had 19 del, and then he received gefitinib, and he got to had T790M to got the resistance for gefitinib, and then he received osimertinib, and he had 19 del and T790M, or 19 del and T0M plus G796S. We notice that all these mutations are not located on the same cell, as they are not located in cis. You could see that they are not on the same reads, so they may be in the different cancer cells. So we draw this. The cancer has very different evolution. And if there have any possible overcome strategies for this mutation of functionality, no. For the 79S in cis with T790M, bractinib and certezumab may work for this, but for the other mutation, no currently targeted therapy is available. So this is the brief introduction of our oncology solution and some cases. Thank you for your listening. And if you have any question, please ask. Thank you.